Hi, everyone. My name is Hae-young Lee, and I'm assistant professor in the Department of Cellular and Integrative Physiology at UT Health San Antonio in Texas, USA. Very nice to meet you, and thank you so much for inviting to um, give me a chance to talk about our research. Although our research in the cerebellum is very mere yet and is not completed, there's really no long story about it, but I really thought that it might be very interesting to share about this with you all guys because the primary cilia deficit in Fragile X syndrome has really been not known yet. We just had a really fresh publication last year in Dentate Gyrus, and that's it. So we wanted to kind of introduce about this and then get your feedback, and then I hope that we can promote this more like a discussion later. Okay, the title of this talk is The Primary Cilia Deficit in Cerebellar Bergmann Glia of the Mouse Model of Fragile X Syndrome. So what is Fragile X syndrome? First of all, Fragile X syndrome is the most common known monogenic cause of autism. And this gene is X chromosome linked. So this disorder is automatically X chromosome linked disorder. And it's caused by a silencing of a one single gene called FMR1, which is Fragile X mental retardation 1 gene. So this FMR1 gene has a promoter with a trinucleotide repeat, CGG repeat. And if there's more than 200 repeats, and then it causes a silencing of the gene. And because this gene expresses very important protein called FMRP, which is an mRNA binding protein, by silencing the gene, FMRP is totally gone, and then FMRP loss can cause a lot of different deficits in whole body. And especially in the brain, FMRP is very abundant throughout the brain. So it can cause intellectual disability, as you can see here, also physical symptoms. But most importantly, 20 to 30% of the patients shows autistic behaviors, such as repetitive behaviors and the social deficits. And sadly, there is no treatment for the core symptoms of Fragile X syndrome. So my lab actually has two major questions about Fragile X syndrome. In general, we are interested in autism spectrum disorder, but we are actually focusing more on the Fragile X syndrome to understand other neurodevelopmental disorders also. And the first question is, what is the cellular and the molecular underlying Fragile X syndrome? So what can we found about the cellular and the molecular mechanism? And once we find that, can we actually develop or therapeutic or the tool so that we can contribute to any treatment of the Fragile X syndrome? And as an effort, we also developed the CRISPR non-viral delivery, and then we have some publications about it. And today I am going to specifically talk about the primary cilia, but we also have the other topics going on in the laboratory, not only Fragile X syndrome, not only primary cilia, but with the other topics too. So coming back to today's topic, primary cilia deficit in the mouse model of the Fragile X syndrome is raised from our lab primarily because we found that primary cilia is very important for the brain development and the neurodevelopment, and then Fragile X syndrome is also the neurodevelopmental disorder. So how can we connect that was our major goal and the major aim of the studies. The primary cilia itself is a sensory organelle that elongates from the cell surface of the most mammalian cell types, including in the brain cells. But most of the cell types actually have a primary cilia, but it's immotile and very small. So if you can see here, this is a cell, and then primary cilia locates usually in the cell body, and then they're about 3 to 5 micron long, so a little longer than the spine in the dendrites. And it is required for multiple signaling pathways, such as sonic hedgehog signaling and then the other pathways. And as you all know, the sonic hedgehog signaling is very critical for early neurodevelopment. So as you may have heard about it. The ciliopathies are the disease caused by the primary cilia deficits. It can be number, it can be length, and then the very representative ones that you might have heard about was a Gilbert syndrome or Bardet-Biedl syndrome. And as you can see here, cerebellum size itself is small, or they're not really properly developed mainly because of the sonic catch-up signaling. So today, we want to focus on the cerebellum development and the primary cilia. So I wanted to bring new attention to the specifically primary cilia loss in the cerebellum development. How does primary cilia loss affect on the cerebellum development? As you can see here, several molecules were knocked out or obligated, specifically or non-specifically in the whole body knockouts by the several papers. And then they found that the loss of primary cilia definitely negatively affect the neurodevelopment in the cerebellum. So as you can see here, cerebellum's lobes are not completely developed, and the size and also the structure is not properly developed in any ages. And other than that, if you look at it really detailed, these molecules by ablating, that it also reduced the sonic catch-up signaling, but not only that, it reduced the granule neuron corpuscle proliferation in EGL, which is transient layer, which was in before birth in human, and very shortly after the postnatal in the mouse, it disappears. It's called external germinal layer. As you all know, this is a very, very important layer for neuronal precursor cells proliferate and migrated into migrate through the Bergman glia later into IGL. So by losing the primary cilia, that people have found that the Gmp proliferation is reduced, and then therefore the EGL thickness is reduced. Especially it was found in mice studies around before the postnatal days P14. The other hand, we want to also look for how can it be related with the FMRP loss. So the cerebellum functions are very defined deficit and impaired in Fragile X syndrome patient, especially not only the intellectual disability, cognitive impairment, but also it has a speech articulation deficit and altered motor coordination deficit and eye blinking condition deficit. So atrophy of the cerebellum lobes were all found in the Fragile X syndrome patients. What about in the FMR knockout mice, which is a mouse model for Fragile X syndrome? In the whole FMR knockout mice, it recapitulates some of the behaviors, cerebellar deficit behaviors is shown in the human patient. So it shows altered motor coordination and deficit in rhythmic automotor coordination, and it also shows deficit in the eye blinking conditions. So we already know that by losing a primary cilia and by losing FMRP, both somehow affect very seriously on the cerebellum function and then development. So how can you connect that? But before then, why did we get this idea, and then why are we looking into primary cilia in the cerebellum? Because we just published one paper. The first brain region that we actually look into was a dentate gyrus in hippocampus. And so long story short, this is kind of a result, the summary of the result that we found. In a wild type, we see the SGG, like a subgranular zone, which is producing the newborn, postnatal newborn neurons, have a cilia normally, but in FMR knockout mice, this postnatal newborn neurons are missing primary cilia. So we got an idea that FMRP somehow affect the primary cilia promotion, and by losing FMRP, FMRP loss cause a primary cilia loss. So we wanted to see how this is going in the cerebellum. So here in this talk, we're going to only talk about the cerebellum, and then we're going to just introduce about our recent observations, which are all unpublished, and then it might need a lot of discussions and feedback from the cerebellum field. Okay, so our major two questions in this talk, and it's going to be straightforward question, is do we see primary cilia deficit in cerebellum in the mouse model of Fragile X syndrome? And if so, does this deficit contribute to the FXS physiology or in using the FMR knockout mice? So as you all know, this is a cerebellum structure, sagittal view. And as you know, the anterior, central, and the posterior view. And if you have this section, you will see this bunch of layers, EGL, ML, PCL, and IGL. Through this layer and then through this anterior to posterior, we were wondering where even primary cilia can have a deficit or are they all normal. So we had to go through almost all anterior to posterior, almost all the layers. And then as you can see here, we measured anterior, we visualized anterior, central, and then posterior by targeting specific lobes. For example, posterior, we targeted the lobe between 8 and 9. And as a result, this might be easier to look at. So as a result, in the Purkinje cell layer, we found the specific reduction of the primary cilia numbers by measuring with the primary cilia marker called ARL13B, and then normalized by the cell number with using DAPI, and then measure the percentage of that. And then as you can see here, there is a reduction in the posterior lobes in the Purkinje cell layer very specifically. So it is interesting that we couldn't really see any other deficit in the IGL or molecular layer or the EGL later on. So I would say that as far as I know, as we observed, we see the primary cilia loss in FMR knockout mice was lobe specific. So it's more posterior side, and the layer specific is more focused on the Purkinje cell layer. And our question is, because Purkinje cell layer has multiple cell types, so we wanted to see and then segregate what cell type is actually missing a primary cilia and then contributing this primary cilia deficit shown in PCL. So as you all know, that we were able to visualize granule cells with a new N and then cal binding on Purkinje for the Purkinje cell, and then BLBP for Bergman glia. And then we did the same thing. We visualized primary cilia using ARL13B and normalized by each cell type markers. So when we counted primary cilia percentage per each cell type, as you can see here, only Bergman glia specifically showed the reduction of the primary cilia, and the other cell types on granule neurons or Purkinje cell didn't show any drastically or significant reduction in FMR knockout mice. So we concluded that the primary cilia loss in FMR knockout mice is cell type specific, and it is specific in the Bergman glia. And we also wanted to know, until now, we only observed in adults. So now we wanted to know if there's any ages specifically causing this primary deficit. So we collected from postnatal day 7 through adult. And for example, postnatal day 7 and 10 and 14 and 28 and adult. At first, we actually did only P7, 14, 20 in an adult, and then kind of have sense that where it started the phenotype, and then we noticed that P14, postnatal 14 days, it started having a phenotype. So we wanted to dissect a little more. So we also did a P10 later on, and then we found that it still started in the postnatal day 10, although we don't know when exactly between postnatal day 7 and postnatal day 10, but it looks like that somewhere between postnatal day 8, 9, 10, this Bergman glia started losing a primary cilia by FMRP knockout mice. And as I introduced, the primary cilia is main role of the primary cilia is mediating the sonic catch-up signaling. So we wanted to measure indirectly how sonic catch-up signalings are affected in this Bergman glia. So what you're seeing here is a Bergman glia in blue. Because we use a GLI1 laxi mice, and then this GLI1 laxi mice, when they're crossed with the FMR knockout mice, we found that the GLI1 laxi positive cells were significantly reduced from again postnatal day 10 and through adults. In this case, we only did for until P30, but we have a supplementary data with also adults. And then this means that the GLI1 is a major protein that which can be indicator for sonic catch-up signaling activation. So we concluded that the sonic catch-up signaling is also reduced along with the primary cilia loss from around P7, which we don't know exact date. And then we concluded that these are all age dependent because we didn't see that deficit when they're younger than postnatal day 7. So our second part of the question, yes, now we see the primary cilia deficit in cerebellum in the mouse model of Fragile X syndrome. So what about does this deficit contribute to Fragile X syndrome pathophysiology? Or what is even Fragile X syndrome pathophysiology? Or what is the symptom shown in FMR knockout mice? So in order to know that, again, this is an unfinished story, so I'll probably just show whatever that we know so far. In the whole knockout of FMR knockout mice, so we see a major symptom as granule neuronal precursor cells proliferation reduction. And that reduction is happening also from postnatal day 10, and that it stays until whenever that we could actually see the granule neuronal precursor cells, which means that EGL, after EGL is gone, we no longer actually can measure this. So we limited our experiment until postnatal day P14. And as you can see here, we labeled proliferating cells with incorporating BRDU during their time for 2 days of injection. And we did the same thing that we visualized a primary cilia and we visualized the whole cell in the EGL layer. And as you can imagine, the EGL layer are very thick when they're born. And then they got thinner and thinner and thinner. And then around the P14, they're too thin. And after P15 or around P16, they're almost gone. So as you can see here, this is a one lobe. And then P10 is actually, it doesn't really have a thick or enough EGL. So these are 2 lobes. And then P14, these are also 2 lobes. So we measure the thickness of this each lobe, and we also measure the proliferation rate of these granule precursor cells. And at the end, we also measured the primary cilia in this EGL just to make sure that we are not seeing, or do we see the deficit in primary cilia itself in the granule precursor cells. Okay, so going to the result, what we found was very shocking because we found that the granule precursor cell proliferation was decreased by the time and in FMR knockout mice. And this is making sense because a lot of people, there are a few reports suggesting that the Bergman glia itself mediate the proliferation of EGL, and then which was shown by multiple groups. So we thought that the Bergman glia missing primary cilia somehow can affect the proliferation of granule neuronal precursor cells, and then this reduced GMP proliferation. And it also results in the EGL thickness, of course, because there is not much proliferation. And to be fair, we wanted to also check the EGL cilia number because is it because of the sonic Hitchcock signaling reduction in EGL itself? Because there are a bunch of reports that EGL cilia is important for the EGL proliferation, I mean granule cell proliferation, but that was not the case when we measured EGL cilia numbers, that we didn't see any reduction in the cilia numbers in the EGL layer. So we wanted to say that this is because this is due to the sonic Hitchcock signaling reduction, and then primary cilia reduction in the Bergman glia somehow affect on the granule cell layer, I mean granule neuronal precursor cells. So in order to prove that, we wanted to obviate FMRP in the Bergman glia, and wanted to check the same thing that what we just found. So basically, from the birth, we injected tamoxifen and using the glass-Cree mice, and then crossed that with FMR knockout mice, conditional knockout mice. And then we injected the tamoxifen in the day 1 and day 2 to induce the knocking out. And then later on, we still injected a BRDU to incorporate into the proliferating cells and collected in the postnatal day P14, postnatal day 14. And as you can see here, these two are the control that which doesn't have a glass Cree yet, but this is a glass wild type, with the wild type for FMR knockout mice, and then this is FMR knockout mice, but these both has FMRP because it doesn't have a glass Cree yet. And this is a control for our FMR knockout mice, which has a glass Cree gene, and the last group has a glass Cree gene with FMR conditional knockout. So this is a true knockout for Bergman glia. So basically, we had 3 controls, and the last group is what we are knocking out, the FMRP, specifically in the Bergman glia. So we measured FMRP level just to make sure that we are seeing is knocking out specifically in the Bergman glia. And then we found that the Bergman glia FMRP intensity is significantly reduced in the last group, as I mentioned. And then these 3 are all control groups. And we also wanted to make sure that this is more specific for Bergman glia, not affecting the Purkinje cells. And then we measured FMRP intensity in doing the same thing in the Purkinje cells. And then you see that there's no difference, significant difference or reduction in FMRP levels in the Purkinje cells. So now going into all the assays that we have done earlier, so repeating exactly the same thing. So now you can see that FMRP ovulation in Bergman glia result in the primary cilia reduction again in the Bergman glia. So that means that the whole knockout losing the primary cilia in Bergman glia is now recapitulated in this glass Cree mice, which is resulting in FMRP ovulation in Bergman glia. So as you can see here, the next, what we did was measuring the GMP proliferation and then measuring the EGL layer thickness. And then we also, to be fair, we wanted to check the EGL cilia number. And as you can see this graph, the GMP proliferation was still reduced, and it was recapitulating the whole knockout phenotype, and the EGL thickness was also reduced and recapitulating the whole knockout phenotype. So lastly, we also show that the primary cilia numbers were not changed in the EGL, so that they support our conclusion that the whole knockout mice phenotype that we have seen for GMP proliferation and EGL thickness was recapitulated by just knocking out FMRP in the Bergman glia. So to summarize in our last slide, that primary cilia loss in FMR knockout mice is lobe specific, and it is more toward the more specific for the posterior lobes. And then it is also layer specific, so it is shown only in the Purkinje cell layer. And it was a cell specific because we only saw that in the Bergman glia in the PCL, and it was age specific because we have observed this mainly from postnatal day 10, but not before a postnatal day 8. And lastly, what we did was Gmp proliferation reduction in whole knockout, FMR knockout mice, was recapitulated by obliterating FMRP mainly in the Bergman glia. So in our study, we wanted to say that this is a good start just to see the primary cilia role in the Bergman glia and how they affect our Fragile X syndrome pathophysiology. So we understand that there are a lot of things to go. For example, can we promote the primary cilia and then see if that rescues any of the genes so that we can really know the causal effect of this? Other than that, these are the people that who mainly work on this primary cilia project. And Bomi Lee is my former postdoc, and Yan Tuma is my current postdoc. Indeed, actually, he's from Czech Republic, and he's doing really well. And the Laura Bowler is a high school student, but he now graduated and go to the undergraduate school. And this was our before pandemic, and the Bummy is sitting here very happily. But now during this pandemic, we have all the meetings through online, and then Yen looks very sad. Hopefully, this pandemic goes away, and that we are all meeting in person in the future. But I think science still goes. And then thank you for all this funding. Although after this pandemic, we actually got the R01 NIH funding, so we're really happy to start again. Thank you so much.